The last of the, well, for me, the last of the macromolecules, we touch on nucleic acids as well. But from a dietary standpoint, carbs, fats, and proteins, those are the three that we talk about. Proteins is the last one. Again, we'll talk about DNA because of, nucleic acids because of DNA and its importance in that regard. But what is a protein? Protein is just a chain of amino acids. Do I have it up here? These amino acids are strung together by peptide bonds. Okay. There are a number of them. They have an amino group, nitrogen, carboxyl group, and a functional group. And it's that functional group that gives it its special sauce. There are 20 of them. I'm not getting into the 20. Some are essential, some are non-essential. Some amino acids, some proteins the body can make, some proteins the body cannot make. Some of them have polarity, some of them have various different charges, which gives them some slightly different functions, but their big purpose their most major function is to make things. Make stuff. I know, technical, right? And the way these amino acids are connected together is all predetermined by DNA. Oh, that pesky DNA, right? So it's that DNA that tells which amino acids to go together. And it's the string of those amino acids that give proteins their complexity. Okay. As these amino acids get strung together, and these are just some examples of some, I'm not going to ask, but they all have a functional group, which is in yellow. Okay. They all have an amine group, which means it has a nitrogen. Okay. And there's your carboxylic group. Okay. Carboxylic acid right there. All of them have that. But again, it's that R group that gives it its special purpose. All right. There's peptide bond. Okay. There's peptide bond. It's the connecting of these individual amino acids that can give proteins their really complex shape. And if you had a certain instructor, he probably dove headfirst into the deep end and talking about some of the major structures of these things. I'm just going to mention them in passing. Okay. We have primary, we have secondary, we have tertiary, and we have quaternary structure. And here you go. Primary, secondary, right there, and right there. Tertiary, third, quaternary, fourth level. All right. The primary structure of amino acid is just the string of amino acids. That's it. Peptide bond interaction, boom. As this peptide chain keeps getting stuff added to it, it gets more longer, and it's going to start twisting and bending and turning around. Okay. When it starts to do that, when it starts to kind of bend or starts to kind of twist around in its structure, then it becomes secondary structure. And that's the beta pleated sheet, and that's the alpha helical nature of it. Okay. Alpha helix, beta pleated sheet, those are all secondary structured proteins. Okay, it's the hydrogen bonds, as you can kind of see there, interacting between the adjacent amino acids that give it that folded appearance or that twisted curled appearance. As we stick a number of these secondary structures together, it can become a tertiary structure. And most proteins, guys, I'm going to tell you, just stop right here. Not every structure, not every protein has to be sequenced into a quaternary structure. But in this tertiary structure, you can see, I mean, there's a number of helix and folds all interacting with one another. And that's what makes that tertiary. Okay. And gives it that 3D shape. Again, and it's the interaction between these secondary structures now that are going to help kind of give it that very three-dimensional complex shape. Okay. That tertiary structure. If I get a number of tertiary structures interacting with each other to form stuff like hemoglobin, okay, then we have our quaternary structure. Okay. Hemoglobin, insulin, okay. Those would be some common examples of that very upper level quaternary structure of a protein. And again, like I said, not every protein goes into that quaternary level of formation there. Okay. There you can kind of see there's some ionic bonds, there's some hydrogen bonds, there's some disulfide bonds, there's even some van der Waal forces. There's a number of things that go in to getting that three-dimensional structure. Am I going to ask that maybe the choice question? Again, I don't really want to do a deep dive into that because I have some stuff coming up in chapter 4 and 5 and 6 that I feel like are more germane to what exactly we're doing here in this class. This is just basic building block stuff that you took last semester, hopefully. Shockingly, you can have many of these macros come together. Proteins and lipids, lipoproteins, proteins and carbs, glycoproteins. Okay? These help to communicate. Glycoproteins, they're that sugar coat, that glycocalyx that you see on the outside of cells. And it's communicating, saying, hi, I'm you. Don't attack me. Lipoproteins can be there to help as receptor sites to bring things into a cell or to get the cell to start making things. So sometimes these proteins like to combine with other biomolecules, and carbohydrates and lipids are those two that they love to dock up with and form structures. And again, these are things that we will see in the plasma membrane of a cell. And we'll hint at some of this stuff next week when we start talking about a cell. So you can see proteins have a huge varied function. You can see number 1 up there, structure. That's probably the most important one, followed by enzymes. If you're going to remember 2, I hope you remember that at least for this first exam, because those are the things that we're talking about in this first exam. Antibodies and carriers and receptors. We'll talk about a little bit of receptors in next week. We'll talk about antibodies and receptors and carriers in Unit 2 and Unit 4. Okay. So Unit 5 as well, sorry. So some other functions, obviously antibodies in the immune system.